नमस्कार दोस्तों, बरकतुल्ला यूनिवर्सिटी भोपाल में पीएचडी एंट्रेंस टेस्ट का सिलेबस क्या होगा उस संबंध में देखते हैं। और जो बरकतुल्ला यूनिवर्सिटी में डीईटी डॉक्टोरल एंट्रेंस टेस्ट के लिए सिलेबस क्या रहेगा उस संबंध में। तो सबसे पहले देखते हैं इसमें बरकतुल्ला यूनिवर्सिटी भोपाल एमपी approved by the coordination committee in its 92nd meeting as per clause 9A of new ordinance number 11 subject microbiology। तो माइक्रोबायोलॉजी जो सब्जेक्ट है, इससे जो पीएचडी करना चाह रहे हैं तो जो एंट्रेंस टेस्ट होगा पीएचडी एंट्रेंस टेस्ट के लिए माइक्रोबायोलॉजी का जो सिलेबस है एंट्रेंस के लिए, उसका डिटेल हम देख लेते हैं। जैसा कि आपने देखा इसमें लिखा हुआ है सब्जेक्ट माइक्रोबायोलॉजी। और आइए डिटेल देखते हैं इसमें। मैक्सिमम मार्क्स 100 नंबर्स, टाइम 2 आवर्स। ठीक है, तो 100 नंबर का जो है टोटल पेपर होगा और 50 नंबर की आपकी रिसर्च मैथोडोलॉजी होगी और 50 नंबर का सब्जेक्टिव रहेगा। यानी टोटल 100 नंबर रहेंगे और 100 क्वेश्चंस भी रहेंगे। हर एक क्वेश्चन के 1 नंबर रहेंगे। ठीक है, और कोई भी नेगेटिव मार्किंग नहीं होगी, सभी ऑब्जेक्टिव टाइप क्वेश्चन होंगे। तो आइए देखते हैं और डिटेल। नोट, सिलेबस डिवाइडेड इन 2 पार्ट्स कैरीज 100 मार्क्स ओनली। फर्स्ट पार्ट ए रिसर्च मैथोडोलॉजी 50 मार्क्स। सेकंड पार्ट बी रिलेटेड टू द सिलेबस ऑफ द सब्जेक्ट। And third, each part A and B will be divided into 5 units each. Fourth, syllabus will be equally divided among all units. And fifth, there shall be no negative marking. अब आते हैं now the part A, and that is research methodology. And maximum marks 50 for this. Doctoral entrance test DET will have question paper in 2 parts. Part A having 50 objective type questions and part B having 50 objective type questions from individual subject of the department. Candidate must score 50% marks to qualify for the interview. ठीक है, अब यहाँ पर maximum marks 50. Part A common for all life sciences. This microbiology comes under life science, ठीक है, कैटेगरी में। So common for all life sciences जो part A है, common है। There shall be 50 objective type question for 1 mark each. Unit 1, basics of research, research methodologies, research topic selection, literature review, etc. Research topic, problem formulation, research and sampling, design, collection and organization of data, types of research, descriptive versus analytical, applied versus fundamental, quantitative versus qualitative, conceptual versus empirical, data collection and analysis, exclusion of research, observation and collection of data, methods of data collection, sampling methods, data processing and analysis strategies, data analysis with statistical packages, hypothesis testing, generalization and interpretation. And now the unit 2nd, research and ethics, definition and overview of research, need of research, research and proposal goals, unethical research practices, code of conduct and legal issues, ethical practices and principles, role and responsibility of research ethics committees, Second, ethics in research execution, importance of consent in research, fundamental rights and moral principles, informed consent, implied consent, explicit consent, active consent, passive consent, written and oral consent, privacy and confidential, confidentiality, confidentiality. And now unit third, basic statistical definitions and concepts, variables and variation, frequency distributions and cumulative frequency distributions, sample and production, center of data distribution and spread of data graphics, histograms, graphs. Then probability, the binomial and normal probability distribution, basic probability distributions, the binomial distribution, continuous data distribution and log normal distribution, choosing samples, random sampling, other sampling procedures, stratified, systematic and cluster sampling, sampling in quality control. Next is a statistical inference, estimation and hypothesis testing, statistical estimation, statistical hypothesis testing, comparison and variances in independent samples, test of equality of more than two variances, confidence limits for a variance t-test. Then unit 4th, introduction to computer and their applications, concepts of operating system, software and database management system, basics of internet and emailing, Microsoft Office, computer networks and internet, flap, http, www, introduction to neural networks, exploring various websites, search engines for collection of quality literature and secondary data, introductory bioinformatics, scope, application and challenges of bioinformatics, useful bioinformatics websites, introduction to genetic algorithms, retrieval of biological data, INTERJ, SRS and DIVJET, LINKUP, database searches, FOSTA and BLAST, sequence filters, iterative database and search and SciBlast, sequence alignment, methods and applications of gene and protein families, methods and applications of phylogenetic trees, phyla, etc., genomics and proteomics, whole genome analysis, comparative genomics, paralogs and orthologs, second generation sequence and analysis, structure prediction and 2D analysis of protein, introduction to drug designing, primer designing. Unit 5th now, bioenergetics, first and second law of thermodynamics, concept of enthalpy, entropy and free energy, high and low energy phosphate compounds, oxidation reduction reaction, activation energy, ATP and its hypothesis, types of bonds. The next is buffers. The Henderson Hasselbalch equation, titration and buffers, physiological buffers, bicarbonate buffer, protein, amino acids and buffer systems. Principle and applications: microscopy, chromatography, electrophoresis, centrifugation, spectrophotometry and colorimetry. Then radioactivity, disintegration of radionucleosides, disintegration of radionucleosides, half-life of radioactive compounds, measurement of radioactivity, scintillation counting, isotopic tracer technique, auto radiography, use of isotopes in vivo and in vitro, labeling, radio diagnosis. Next is the techniques used in recombinant DNA technology is cloning vectors, cloning in bacteria and eukaryotes, construction and screening of genomic and cDNA library, application of recombinant DNA technology in medicine and agriculture, GMO, application of gene cloning in synthesis of drug and enzyme, insulin, interferons. The next is a restriction mapping, southern blotting, northern and western blotting techniques, RFLP, RADP, RAPD, SSR, 16S, RNA, DNA sequencing techniques, preparation of radio-labeled and synthetic probes, gene amplification, DNA chip technology and microarray. And now for the part B, PhD entrance syllabus for microbiology. There is a 50 marks for this part, and this is means your syllabus which you done the post graduation on that particular subject. And the first unit is general microbiology. In this, first is bacteria, origin of life, structure of bacteria, cytoplasmic and outer membrane, capsule, flagella, pili, endospore and special organelle, gram negative, gram positive and acid fast bacteria, culture of bacteria, aerobic, anaerobic and facultative, isolation in pure culture and its characteristics, nutritional types, culture media, strategies of cell division, growth, kinetics, generation time, synchronous batch, continuous culture, measurement of growth and factors affecting growth, control of bacterial growth, physical and chemical agents, preservation methods, stress responses, study of bacterial growth, kinetic effect of inhibitors and stimulators on growth, characterization of bacteria, shape, gram, stain, endospore, stain, capsule stain, acid fast stain, flagella stain, assay of antibiotic, MIC and MBC microbiological media. Now next is the virus. Nomenclature and classification, morphology and ultrastructure, capsids and their arrangements, types of envelopes and composition, genome, type and structures, subviral particles, viroids, virusoids, prions, lytic and lysogenic cycle. Now this fungi or fungi. General characteristics, somatic structure, classification and method of identification, classical and molecular approach, thallus organization, symbiotic association and parasexual life cycle, asexual and sexual reproduction of fungi, growth and its measurement, mycorrhiza, eco, indo and fourth A mycorrhiza. And now next is algae. Classification, alga pigments, thallus structure, nutrition, ecology, sexual and asexual reproduction and their importance, SCP, biofuels, food, chemical and pharma, pharmaceutically important products. Next unit is this biochemistry. Structure and functions of biomolecules like carbohydrates, lipids, protein, nucleic acids, and vitamins, enzyme kinetics, enzyme inhibition, allosteric enzyme, rate limiting enzymes in multi-step reaction, rise, rhizome and enzyme, ribozyme and enzyme, electron transport chain and oxidative phosphorylation, pathway and regulation of major metabolism, glycolysis EMP pathway, TCA cycle, glyoxylate cycle, interdorov pathway, pentose phosphate cycle, biological buffers, biological transport active and passive, bio signaling mechanism and role in diseases. And now the third unit is cell biology, prokaryotic cells, cell walls, cell membrane, mechanism of solute transport across membranes, flagella and pili, capsule, cell inclusions like endospores and gas vessels, eukaryotic cell organelles, endoplasmic reticulum, Golgi apparatus, mitochondria and chloroplast. And now in a fourth unit, genetics and molecular biology. In this, DNA replication mechanism, protein and enzymes involved, regulation, DNA damage and repair and recombination, DNA damage and repair mechanisms, recombination mechanism, types and biological roles and practical applications, transcription mechanisms, proteins and enzymes involved, regulation, transcription, activator and repressor, cis acting and trans acting elements, RNA polymerase, capping, RHO protein, inhibitors of transcription, protein synthesis and processing, ribosome, translation mechanism, protein and enzymes involved, tRNA structure, amino acid activation, initiation complex, initiation factor and their regulations. elongation factors, termination, genetic code, post translation modification of proteins, inhibitors of translation, regulation of gene expression, operon concept, role of chromatin in gene expression and gene silencing, genomic imprinting, types of mutation, physical and chemical mutagens, selection of mutants, aims test of mutagenesis, bacterial genetic system, transformation, conjugation, transduction, recombination, plasmids, transposons, RNA interference, principles and procedures of protein and nucleic acid, sequencing, southern, northern and western blotting, polymerase chain reaction, RT, PCR, real-time PCR, mutagenesis, gel electrophoresis, nucleic acid sequencing, phyrosequencing, RFLP and RAPD, SNP, VNTR analysis, restriction, enzymes, vectors, gene cloning, gene therapy. And now the fifth unit, immunology, cells and molecules involved in innate and adaptive immunity, antigens, antigenicity, and immunogenicity, epitope and paratope, structure and function of antibody molecules, generation of antibody, diversity, monoclonal antibodies, antigen antibody interactions, MHC molecules, activation and differentiation of B and T cells, receptors, humoral and cell mediated immune responses, primary and secondary immune response, the complement system, inflammation, hypersensitivity and autoimmunity, immunity to infections. Now the sixth unit, medical and diagnostic microbiology. In this, epidemiology, symptomatology, general description of microbial pathogens, endotoxins and exotoxins, diagnosis, prevention and therapy of meningitis, tuberculosis, leprosy, urinary tract infection, cholera, ringworm, syphilis, diphtheria, mycotoxicosis, opportunistic fungal pathogens, dermatophytes, malaria parasite, giardia and lysephamia, types of vaccine, live microorganism, attenuated organisms, genetically modified organisms, protein, edible, synthetic, recombinant and anti-iodotype vaccine. And seventh unit is fermentation technology and industrial microbiology. Types of fermentation, bioreactor configurations, stirred tank, bubble column, airlift reactor, stirred and air driven reactors, packed bed, fluidized bed, tracked bed, monitoring and control of bioreactors, ideal reactor operation, batch, fed batch and continuous operation, downstream processing, filtration, centrifugation, cell disruption and ideal stage concept, aqueous two-phase liquid extraction, adsorption, chromatography, solid state frame fermentation. Eighth unit is environmental microbiology. Environmental complex interaction of ecological factors, light, temperature, precipitation, rainfall, humidity of air, atmospheric gases and wind, topographical factors, edaphic factors, extremophile, anaerobes, halophiles, acidophiles, alkalophiles, thermophiles, barophile, community barophile. Community structure and organization, aeromicrobiology, microbes of indoor and outdoor environment, pathways, enumeration, extramural and intramural control, bioterrorism, euphoification, biosafety, water microbiology, significance of microbes in water quality, test for probability of water, microbial treatment of sewage, application of sewage water in land, compositing of biosolids and domestic solid waste. And the now ninth unit is food and agriculture microbiology, microbial spoilage of foods, fruits and vegetables, preservation of foods, principles of quality control, microbiological safety of food, soil, rhizophores and phyllosphere and microphoria, microbial plant interaction, carbon, nitrogen and phosphorus cycles, integrated pest management, viral control of pathogens. That's all, and please refer if you have any book with you, so you can find out all these topics from that book. But if you don't have any book, so please visit internet. With the help of internet you find all these topics over there and cover all these topics in a very good manner. That's all. बस इतना ही है, मिलते हैं अगले वीडियो में, तब तक के लिए नमस्कार, धन्यवाद।